Hello students. So today we are going to discuss about the NEET 2020 biology paper, and we will be dealing regarding the explanations of the questions that has been asked. All right, and we will be dealing in detail and explaining the questions. That means the answers of the questions in detail. So let's start with the very first question. So the first question says that you have to match the organism with its use in biotechnology. So on your left side, the name of the organisms have been given, and on the right side, their use has been given. All right. So the very first option, which says regarding the Bacillus thuringiensis. So option A, which is Bacillus thuringiensis, you might have been knowing this also, that Bacillus thuringiensis is a very common Bt bacterium, which is prominently used in biotechnology. So this Bacillus thuringiensis is prominently and commonly used as a cry protein. It's actually a source of cry protein, which is the option fourth here, you can say. All right. And these cry proteins are actually used for killing the insects. All right. So this is a source of cry proteins, you can say. All right. So this is the very first important match that we have seen. So the option for A will be fourth. So this will go with A option. Clear? Now, the second option is the Thermus aquaticus, which you can see on your screen. So Thermus is basically, Thermus, the entire name of Thermus is basically Thermus aquaticus. And Thermus aquaticus is a thermostable bacterium which is commonly used in the DNA polymerization reaction, or you can say in the PCR method. That means polymerase chain reaction method of DNA polymerization. So that's why Thermus aquaticus is actually used in the process of DNA polymer. So this is actually a source, just a second. This is a source of thermostable DNA. Thermostable DNA polymerase. Why thermostable? Because they are very much resistant to high temperature. So that's why they are very much, since they are very much resistant to high temperature, so that's why they're a source of thermostable DNA polymerase. And this thermostable DNA polymerase is also known as tag DNA polymerase. Tag DNA polymerase, you can say. Clear? So the option B will lie over here. This will be the option. All right. So A will be for and B will be for third option. All right. Now, the third category says about the Agrobacterium tumefaciens, which you can see. Again, this is also a very common bacteria, which is prominently used in the process of cloning vectors. It is actually used as a, it is actually used in the process of biotechnology for, for transferring of the genetic material or the DNA from one cell to from the host cell, you can say, or from its own cell into the recombinant cell. So we can say that this Agrobacterium tumefacien Agrobacterium tumefaciens. It's tumefaciens. Is a source of cloning vector, or not, we can say not a source, but this is actually a cloning vector. Is a cloning vector. Clear? This will be the case. And the last option is that of the Salmonella typhimurium. So Salmonella typhimurium is basically the first bacterium, the first ever bacterium whose first recombinant DNA molecule was constructed. This one, Salmonella typhimurium. So the first ever construction of the recombinant DNA molecule was actually performed using a native plasmid of the Salmonella typhimurium. So this will involve the option of, so C was the option of 1 and Salmonella typhimurium will be option D. All right. So we can just write the explanation again over here, that Salmonella typhimurium was used for the construction of first recombinant DNA molecule. Clear? So these are the options that needs to be matched. Now let us take a look into the different options. So if you're going to look into the options, so A will be for the fourth option. So A will be here for the fourth option. So here it is coming up, as you can see. And then B will be for the third option here, as you can see over here. Then C will be for the first option, which is this one. All right. And again, the fourth option will be for the construction of the recombinant DNA molecule. That is going to be here. All right. So this is the case. So here mistakenly has been printed 3, but here it will actually be 1. Here it will actually be 1 over here, not 3. So there's a printing mistake. So according to the given option, option A is considered to be as the correct answer. That means option A. Clear? So this is the first question. Now let's move on to the next question, which is going to be the, again, this is also a match the column that has been given over here. So you have to match with the correct options. Clear? Now, out of all the options which you can see, let us take a look into the different categories. So the first option says regarding the inhibitor of the catalytic activity. Here the different kinds of chemical agents have been given, or different molecules actually have been given. So that is the case. So the first option, which is the inhibitor of catalytic activity, so this will match up with the malonate. So this is going to match up with malonate. So A here will be the option. That means it will match up with second one, because malonate is the competitive inhibitor of the catalytic activity of a very important enzyme, and that is succinate dehydrogenase. So we can just write up that the malonate is a competitive inhibitor actually, is a competitive inhibitor of the catalytic activity of a very, very important enzyme, and that is succinate dehydrogenase or succinic dehydrogenase, you can say. Now, this is one such enzyme that is found in the, you know, uh, in the inner mitochondrial membrane of the mitochondria, and that plays a very, very important role in your electron transport system. And this is the only enzyme to be found in the inner mitochondrial membrane. Otherwise, all the enzymes that participates in the this one, which is the process of your citric acid cycle, those are all found in the mitochondrial matrix. But this is the only enzyme which is involved both in the process of your citric acid cycle also and ETS also. So succinate dehydrogenase, you can say. Clear? So this is the case. And the second option says that it possesses peptide bonds. So peptide bond is actually being possessed by collagen, you can say. All right. So out of all the three options that has been left out, so peptide bonds are being possessed by this collagen. All right. So collagen is actually proteinaceous in nature, you can say. All right. It's a kind of a complex polymer which is proteinaceous in nature, and that possesses peptide bond. So you can say that collagen is protein in nature, possessing peptide bond. Peptide bonds. Clear? So two options are already done, this one and this one. Now, cell wall material in fungi. So this is again a very easy question, you can say, or easy option, you can say. So cell wall material in fungi is going to be chitin, this one. We have been studying this part, from very early. So this is going to be the option of C over here, you can say. So chitin is actually a homopolymer that is found in the cell wall of fungi also. And chitin, if you people remember, then this is also found in the exoskeleton of arthropods. That means insects also. All right. So option C will say that chitin is a homopolymer found in the cell wall of fungi and exoskeleton. Exoskeleton means the upper skin, you can say. Exoskeleton of arthropods, which is the largest kingdom of kingdom Animalia. Clear? This is the case. Now, the third option has been done. Now, the fourth option, if you are going to look into, that means secondary metabolite. So the fourth option is left with the first option only. So ricin is considered to be as a secondary metabolite over here. Ricin is actually considered to be as a secondary metabolite. In fact, you can say ricin and heparin. These are actually toxins, and secondary metabolites are those metabolites which are produced in the plant bodies not all the time. All right. So they are only produced in the plant body when it is required by the plant body during certain conditions or specific conditions only, you can say. All right. So you can say that heparin and ricin are the toxin secondary metabolites. So since these are toxins, so these are actually being released by the plant body in the form of defense mechanism. Secondary metabolites. Clear? So this is the case. So we have seen all the four options. Now let us take a look at which option actually matches up. So A option is going to go with 2. So A is going to go with 2 over here, you can say, here, this one. Then B is going to go with your fourth option. So you can see over here. Then C is going to go with third and D is going to go with first option. So our correct answer is option 4, which means this one. This is the correct answer. Clear? Now, moving ahead to the third question. That says that the plant parts which consist of 2 generations, all right? This question says that the plant parts which consist of 2 generations, one within the other, you have to find out, all right? So you have to find out the 2 generations. Now, pollen grains inside the anther, all right? So pollen grains inside the anther and is the first option. Then germinated pollen grain with 2 male gametes, then seed inside the fruit and embryo sac inside the ovule. So out of all the options which you can see, so the 2 generations, here the key point is the 2 generations that you have to note it out. So 2 generations means the first option will be the correct option, all right, because pollen grain is actually haploid and the anther, see, this pollen grain is produced in the anther. What will be the answer? So the answer is basically your option A. Option A will be the answer. This will be the correct answer. This is one of the first answers. Why? Because anther is, these pollen grains are produced inside the anther and we know that anther is diploid. So anther is diploid, which means 2N, while pollen grain is haploid. Pollen grains are haploid. Clear? So this indicates that there are having 2 generations. One is of haploid one and the other is of diploid one. Clear? So the first one will be correct. Germinated pollen grain with 2 male gametes. Now, 2 male gametes are actually found inside this pollen grain and both of them are haploid. This is also haploid and pollen grain is also haploid. So definitely this is not going to be the correct answer because male gametes here, if you are going to just write it out, just a second. You can just, yeah. So here, if you're going to just write it out, then this, male gametes, these male gametes are actually haploid. Male gametes along with pollen grain, they are both haploid. They are both haploid. So we cannot consider this as the 2 generation because both of them are haploid. So that's why the statement is wrong over here. Then seed inside fruit, again, fruit is also diploid and seed is also diploid. So again, this is also a wrong statement. Now, the last statement which is left out is the D option, embryo sac inside the ovule. Now, embryo sac, if we are going to consider, so embryo sac, which is also considered to be as female gametophyte. So embryo sac is also haploid. So embryo sac is haploid and Ovule, if you are going to see, then ovule is diploid because inside ovule, the embryo sac is being found. So ovule is diploid. So again, the two generations are found. So technically, if you are going to see, so there are two correct options. That means A and D, which means option C is the correct answer over here. Clear? This is the case. So this is the third question that we have seen. All right, now let's move on to the next question, which says that the oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of. See, this Rubisco enzyme basically stands for, this is one of the most abundant enzyme in the entire biosphere. This we know this fact, and this is used in the process of C3 cycle or C4 cycle of photorespiration. But here it is asking about the photorespiration. So in photorespiration, basically the process of oxygenation takes place, which means binding of Rubisco to this enzyme is going to bind the oxygen rather than carbon dioxide. All right. So basically we can say that the oxygenation, so the correct explanation will be, the answer will be that the oxygenation of Rubisco, which is ribulose bisphosphate carboxylase oxygenase enzyme, during photorespiration it leads to formation of one molecule of 2-phosphoglycolate. So it leads to formation of one molecule of 2-phosphoglycolate. Phosphoglycolate. Now this phosphoglycolate is a 2-carbon compound. Let me tell you one thing. All right. While along with this molecule, that means one molecule of 2-phosphoglycolate, there is another molecule which is also being formed, and one molecule of 3-phosphoglycerate. And one molecule of 3-phosphoglycerate, which is shortly termed as 3-PGA. Clear? So this is actually a 3-carbon molecule, you can say. All right, so this is the case. So how the reaction goes on, the reaction is something like this. RUBP plus the reaction, you can say, is RUBP is going to undergo the process of oxygenation in the presence of Rubisco over here, and it will be forming one molecule of 2-phosphoglycolate and this phosphoglycolate is actually a 2-carbon molecule, and this is going to enter into a separate pathway which is called as glycoxylate pathway. All right, glycolate pathway, and it is going to form one molecule of 3-PGA, which is 3-phosphoglycerate, like this. All right, so our correct answer, as you can see, out of all the 4 options, our correct answer comes out to be option C, which is one molecule of 3-carbon compound, that is PGA, and one molecule of 2-carbon compound, which is 2-phosphoglycolate. So option C is the correct answer. Now, the next question. Now, the next question says that in relation to the gross primary productivity and net primary productivity of an ecosystem, which of the following statement is correct? All right. So which of the following statements are correct in relation? So this is a question from your ecosystem chapter. So what is gross primary productivity? So if you people know that GPP, the formula, all right. So gross primary productivity is considered to be as NPP minus R. That means net primary productivity minus respiration losses. So this indicates respiration losses. Sorry, just a second. There has been a little bit mistake. I have written the opposite terminology actually. Now, so actually the net primary productivity, just a second. So the net primary productivity is equal to gross primary productivity minus respiration losses. And GPP is actually NPP plus R. This is the formula. That was the actually, I just mistakenly, wrote that part. So basically this NPP is your gross primary productivity minus respiration losses. All right. So this stands for respiration losses. See, net primary productivity is considered to be as the actual amount of productivity which is available for consumption to the consumers, you can say, or to the herbivores, you can say. That is called as the net primary productivity. And this GPP, which is called as the gross primary productivity, this is actually the total amount of, you can say, photosynthetic product which is produced by the producers. All right. So that is the case. So if we are going to just write the definition, so GPP, it is the total amount of photosynthetic product that means the entire, including NPP plus R, product produced by plants. Clear? Now, NPP, if you are going to consider, so NPP is the net productivity that is available for consumption only to the consumers, you can say, that is being assimilated, or that can be transferred to the next trophic level. All right. So you can say that it is the amount of productivity available for consumption by heterotrophs. So this is actually the amount of productivity which is available for consumption by the heterotrophs or the consumers, you can say. So indirectly, we can say, we can also, by this thing, we can also correlate one thing, that GPP is always greater than NPP. GPP is always greater than NPP. So we can just read out the statement that gross primary productivity is always more than net primary productivity. So yes, so this is going to be the correct statement. This is the first statement which we found it to be the correct statement. Then the second statement says that gross primary productivity and net primary productivity are one and same. Now again, this is wrong. Clear? Now there is no relationship between gross primary productivity and net primary productivity. Again, this is wrong. Gross primary productivity is always less than net primary productivity. No, this is also again wrong. Clear? So our correct statement turns out to be option A. Clear? So this is the explanation that we have seen. Now let's move on to the next question, sixth question. So this says that the product of the reactions which is catalyzed by the nitrogenase enzyme, all right, in the root nodules of leguminous plants is or are. So products, you have to see the products which is being catalyzed by the nitrogenase enzyme. Now nitrogenase is one such enzyme that fixes nitrogen, all right, and converts it into ammonia and hydrogen. So if I just simply write down the reaction for nitrogen fixation, so the reaction turns out to be N2 plus 8H plus 8 electrons. Along with that, 16 molecules at the expense of the 16 molecules of ATP, and in the presence of enzyme nitrogenase, along with magnesium ions, this is going to give rise to 2 molecules of ammonia along with 16 ADP plus 16 pi plus 1 molecule of hydrogen over here. All right, 1 molecule. So this is the entire reaction of the nitrogen fixation that you can see. So what if we just look into the options, then is it nitrate alone? Again, we haven't got any nitrate because nitrate is being indicated by NO3 minus. And we get actually nitrate in the process of nitrification. All right. Then there is ammonia and oxygen. No. In this process, nitrogenase will get irreversibly inactivated in the presence of oxygen. So if there is oxygen, this nitrogenase enzyme will get irreversibly inactivated. So there has to be completely anaerobic environment for fixing the nitrogen into ammonia or fixing the nitrogen in the root nodules of the leguminous plants. Clear? Again, so this is also wrong. Ammonia and hydrogen. So we have got ammonia also as one of the product and hydrogen also as one of the product, which you can see. So this is ammonia and that is the hydrogen, as you can see. So this is the correct answer. Clear? And ammonia alone? No. So our correct option is option C. Clear? So this is the 6th question. Now, next move on to the next question. So the next question says that you have to identify the incorrect statement with the 4 statements that has been given over here. So the first statement says that sapwood is involved in conduction of water and minerals from root to the leaves. All right. So that is the case. And the next statement says that sapwood is the innermost secondary xylem and is lighter in color. All right. Due to deposition of tannin, resins and oils, heartwood is darker in color, and heartwood doesn't conduct water but gives mechanical support. So these are the 4 statements that you can see. Now, let me tell you the incorrect statement first. So the incorrect statement here is the statement B. Incorrect statement is the statement B because there are 2 types of wood which are being found. Now, let me tell you one thing. 2 types of wood are being found. One is called as the heartwood and the other is called as the sapwood. So heartwood and sapwood. So this basically represents, both of them represents the secondary xylem only. They are actually the component of secondary xylem only. But one of them is responsible for conduction and the other is not responsible for conduction of water and minerals. All right. So in this category, you have to identify the incorrect statement. So the incorrect statement is that this sapwood is actually found in the outermost secondary xylem and it is usually darker in color. All right. Sorry, lighter in color. So it is lighter in color, but it is found in the outer area. All right. So the incorrect statement is going to be the incorrect statement turns out to be option B over here, which says that sapwood is innermost secondary xylem. So this is not the innermost secondary xylem, but this is actually the correct statement will be, it is the outermost secondary xylem. So sapwood is outermost, or you can say outer, actually, secondary xylem. Clear? This is the case. Otherwise, this lighter is correct, but all other statements are clear. That sapwood is involved in the conduction of water and minerals. Like this is also true, because since this is the outermost part, so that's why it is involved. And due to the depositions of tannins, resins, and yes, heartwood and sapwood, they are entirely opposite to one another. Heartwood is not involved in case of conduction, but the sapwood is involved in case of conduction. Heartwood actually gives mechanical strength. So this is again, this is also true. All right. So the incorrect statement regarding to this will be the option B1. Clear? So this is the case. Now, next question. Now, BT cotton variety that was developed by the introduction of toxin gene, all right, that is Bacillus thuringiensis, that is resistant to. All right. So this is again an important question that has been asked from your biotechnology section. Now, let me tell you that this BT cotton is actually resistant to cotton bollworm, which is a category of insect pest. So the correct option here will be insect pest. And why insect pest? Now, let me tell you, because BT cotton is resistant to cotton bollworms. And for that part, various kinds of cryproteins have been, found out. So cotton bollworms, all right, which is a category of insect pest. Clear? Like, for example, as I already told you, that cryproteins genes have been introduced in the cottons to protect it from any kind of diseases. So what are those proteins? So those proteins are cry1AB and cry1AC. Sorry, cry2, cry1AC. Just a second. It's going to be cry1 AC and cry2AB. Cry basically are the cry proteins. These are a category of proteins. So cry2AB. This is the case. Now these genes have been introduced in cotton to protect it from bollworms. And that's why BT is also considered to be as a biopesticide. So you can say that that's why BT cotton acts as biopesticide. All right. So with this case, our correct option will be option D. That means insect pest will be there. Clear? Now, next question. Now, which of the following pairs is of unicellular elega? Now, you have to find out the category of unicellular elega among the given options. So the very first case is Gelidium and Gracilaria. So in case of Gelidium and Gracilaria, now let me tell you that this Gelidium and Gracilaria, they are the category of Redalegi. Gelidium, and Gracilaria. They are the category of red algae, and they are not unicellular. They are not unicellular because they all are multicellular. And they are multicellular category. Clear? This will be the case. While the option B says, so this will be not an option. So the second option is Anabina and Volvox. So Anabina and Volvox, if you are going to consider, here we have to underline it since these Gelidium and Gracilaria, they are genus. So Anabina is a category of blue-green algae, which is BGA, while Volvox is an example of colonial green alga. So this is not unicellular, all right. So this is colonial green alga. That means Volvox. Clear? And this Anabina is actually filamentous. This is filamentous algae, blue-green algae. So again, this is also not an option. Then the third option turns out to be chlorella and spirulina. Now, this is supposed to be the correct answer because this chlorella and spirulina both are the unicellular green alga and spirulina. Both are unicellular green alga. So this is the correct answer. And the D option again, D option is Laminaria and Sargassum. So Laminaria and Sargassum both are multicellular, multicellular, this one, brown algae. It's Laminaria and Sargassum. So these are multicellular brown alga, or brown algae you can say. Both. All right. So again, this is not an option. So our option C is the correct answer, chlorella and spirulina. All right. So this is the 9th question. Now let's move on to the next question, which is question number 10. Strobili or cones are found in. All right. So strobili or cones, if you people remember, so these are the structures which are found in case of pteridophytes and gymnosperms. All right. So these are actually the reproductive organs. So strobili or cones are actually the reproductive organs in case of pteridophytes and gymnosperms, you can say. You might have heard about male strobili and female strobili. So strobili is actually cone-shaped like structure where the sporophylls are arranged. That means leaf-like structures are being arranged. So reproductive organs in pteridophytes and gymnosperms. Clear? So they are prominently found in. All right. Now, since these strobili and cones, they are not found in all categories, but yes, we can say they are actually found in some of the selective categories. So the options given are Pteris, Mercantia, Equisetum, and Salvinia. So out of all these options, which you can see over here, all right. So the correct option will be Equisetum. All right. So strobili or cones, they are prominently found in case of equisetum, which is a higher category of pteridophyte, you can say. All right. So this is the part. Now let's move on to the next question. So this says that name the enzyme that facilitates the opening of DNA helix during the transcription process. Now the opening of DNA helix, all right, it is talking about, remember, during the transcription process. Transcription means a process in which the DNA will be converted into mRNA. All right. So the enzyme which is responsible for this is whether it's DNA helicase or whether it is DNA polymerase or whether it is RNA polymerase or it is DNA ligase. So opening of the DNA helix is being promoted by your DNA helicase enzyme. All right. So the correct answer will be option A. DNA helicase actually facilitates this is option A, all right, facilitates the opening of DNA helix. Clear? This will be the case. Now, this option B, DNA polymerase will be responsible for polymerizing the DNA, for the DNA replication process. So B, which is DNA polymerase, this is for DNA. This is actually required in, you can say, DNA replication enzyme, DNA replication process. So required in DNA replication. Clear? Then RNA polymerase is required in polymerizing the RNA. Later on, it is being required. So that means in the process of transcription, but not in the opening of the DNA helix. All right. So RNA polymerase is actually required in transcription process for synthesizing mRNA, you can say. Synthesizing mRNA, which is messenger RNA, and the D option, which is DNA ligase. DNA ligase is used. DNA ligase is actually a kind of a sticky enzyme, you can say, and this is used to seal off any kind of breakage in the DNA strands. So that is called as DNA ligase. So this is a sealing enzyme that is used in DNA replication. In DNA replication. So the correct answer will be option A. Clear? This will be the case. Now, the next question says that with reference to the wrong, identify the wrong statement with reference to the transportation of oxygen. Now, with reference to the transportation of oxygen, you have to identify the wrong statement, and this is a question from your respiration chapter. So the first statement says that the partial pressure of carbon dioxide can interfere with oxygen binding with hemoglobin. So the first statement is partial pressure of CO2. Does it interferes with the oxygen binding of hemoglobin? Yes. This is a correct statement, because if the partial pressure of carbon dioxide will be greater, if this is, we can like do like this, wait. So if partial pressure of carbon dioxide is higher than partial pressure of oxygen, then definitely the binding of the binding of carbon dioxide will be taking place with that of the hemoglobin. So then CO2 binds with hemoglobin, binds with hemoglobin, you can say. All right. So definitely it is going to interfere. So this is the correct statement. All right. Now, higher hydrogen ion concentration in the alveoli favors the formation of oxyhemoglobin? No, this is a wrong statement. Higher, a higher concentration of this, this hydrogen ion concentration in the alveoli is not going to promote, or it is not going to favor the formation of oxyhemoglobin. It is actually going to go for the dissociation of the oxygen with that of the hemoglobin. All right, so you can say that because higher the concentration of oxygen, then this indicates that greater will be the PO2 availability, and which will actually favor the formation of oxyhemoglobin. So again, this is a wrong statement. B statement is the wrong statement that we have seen. So we can say that higher, hydrogen ion concentration, if there will be higher hydrogen ion concentration, then this will result into low binding of oxygen with hemoglobin. Clear? So this is the wrong statement. So we have found out the wrong statement, but let us look into the other statement as well. So the next statement is that low PCO2 in the alveoli favors the formation of oxyhemoglobin. Definitely. This is also a correct statement, because if, as I told you, that if the partial pressure of carbon dioxide will be less, all right, then it will favor the formation of, or favor the binding of, then oxygen will bind with hemoglobin. So again, this is also a correct option. And the D option says that binding of oxygen with hemoglobin is mainly related to the partial pressure of oxygen. Yes, again, this is also a crucial statement. Yes, that the partial pressure of oxygen determines the binding of oxygen with hemoglobin, you can say. Clear? So the wrong statement turns out to be option B. So the correct answer will be option B. That means which is actually the wrong statement. All right. So let's move on to the next question, which is 13th. Now you have to identify the correct statement with regard to the G1 phase of interphase. So if you people remember, then this phase, that means G1S and G2M. So the sequence cell division phases, if you are going to see, so this is G1, then G2, sorry, it is G1 and then S. So G1, then S phase, then G2, and then M phase. That means mitotic phase, the phase of mitosis. So these three are the phases of interphase. These are the interphase. All right. Now, so you have to, talk about the correct statement regarding the gap 1 phase of interphase. Clear? Now see, so this says that reorganization of all cell components takes place. That means the reorganization of all cell components takes place. So uh, reorganization means that all the cellular components, they will be reorganized. So this kind of a statement is a little hazy, you can say. So this is not as such correct, but we are going to still look into this statement. All right, so actually this is a wrong statement. So cell is metabolically active, grows and does not replicate its DNA. Yeah, so this we can say that this is the correct option, because in case of gap 1 phase, cell is actually active, and as soon as this G1 phase is preceded by, this is followed by this S phase, which is the phase of DNA replication. So S phase is the phase of DNA replication. So here the cell will be actually metabolically active, and it will synthesize all the requirements, all the proteins, enzymes that will be actually required in DNA replication. So here the cell is, you can say, is metabolically active. Clear? So we can say that this is a correct statement. Now nuclear division takes place? No. Nuclear division takes place in M phase. Here the process of nuclear division takes place. Nuclear division. Clear? So this is a wrong statement. DNA synthesis or replication takes place. Again, this is also a wrong statement, because DNA replication takes place in S phase that I have note down over here. So our correct answer comes out to be the option B, which is cell is metabolically active, grows but does not replicate its DNA. Now let me tell you that this part, that means reorganization of all cell components, this also occurs, as I already told you, that all the cellular components, that means all the cell organelles and all those, processes of division and all those things. So here the nuclear division also takes place, and here the reorganization also takes place. So our correct answer will be option B. All right. Now, next question. Which of the following statements about inclusion bodies is incorrect? All right. So inclusion bodies are one such bodies that are found inside the cells, and that have very specific functions, or very specialized functions, you can say. All right. So let us look into the incorrect statement. Actually, you have to find out the incorrect statement regarding the inclusion bodies. So are the inclusion bodies involved in ingestion of food particles? Yes. These inclusion bodies are actually involved in the ingestion of the food particles. So this we can say is the correct answer. They lie free in the cytoplasm? No, they are not lying free in the cytoplasm like this. So this is a wrong statement, you can say. They represent reserve material in the cytoplasm? No. The reserve material is being represented by different cell organelles. All right. So again, this is a wrong statement, and they are not bound by any membrane. So again, this is also wrong. Inclusion, in case of the inclusion bodies, they are actually bounded by membrane. So we can say that one of the most important properties of inclusion bodies is to be involved in ingestion of food particles. Just a second. I have seen this statement to be as the incorrect statement. So we have to find out the actually incorrect statement over here. I have seen the correct statement. So we are just going to, just a moment. There's a little confusion. We're going to do this question again. Sorry for the inconvenience. So we have to actually find out regarding, the statements about the inclusion bodies, which is actually incorrect. All right. So we have to find out the incorrect statement, and I have considered it earlier to be considered it to be the correct statement. So that's where the confusion happened. So basically, which of the following statements about the inclusion bodies is incorrect? That is, it is talking about. Now, inclusion bodies are actually, they lie in the, they are involved in the ingestion of the food particles. Now, they are actually not involved in the ingestion of food particles, because in the ingestion of food particles, the organelles which are involved or the cells which are involved, they are called as phagocytes. They are considered to be as phagocytes, which are involved in the ingestion of the food particles. All right. So again, this is the incorrect statement. So with this aspect or respect, the incorrect statement goes on. All right. So these inclusion bodies, they lie free in the cytoplasm. This is correct. They represent reserve material in cytoplasm. Again, they are going to store food. Yes. Then they are not bounded by any membrane. Yes. Again, this is also correct. So the incorrect statement with respect to this will be the option A, because ingestion of food particle is being performed by the phagocytes. All right. So this will be the correct thing. So you have to be very precise in answering all those things. Clear? Now, moving ahead. Now the next question is that again, match the following diseases with the causative organisms have been given, and you have to find out the correct statement. All right, now let us take a match. So typhoid. All right, so the first option is typhoid. Now typhoid is caused by a bacterium which is known as Salmonella typhi. So this is caused by Salmonella typhi. Clear? So this will go with option A. Clear? Now, pneumonia is caused by the genus which is called as Streptococcus pneumoniae. So this is Streptococcus pneumoniae. This is the case. Clear? Streptococcus pneumoniae also, and Haemophilus, influenzae also. Basically, there are two species actually. So that is being involved, both of them. And so this pneumonia will go with Streptococcus has not been given, but Haemophilus is being given. So this will go with option B. So this, and it is also caused by Haemophilus influenzae. Clear? This is the case. Now, option C says filariasis. Now, filariasis is actually caused by, filarial worm. Now, this filariasis, which is also known as elephantiasis, all right. So, this is caused by a worm, and that is called as Wuchereria. And there are two species of Wuchereria. So, it is filariasis is caused by Wuchereria. And there is Wuchereria bancrofti and Wuchereria malai. These are the two species which causes filariasis. So this will go with the option of C. Clear? And then the last comes out to be malaria, which is very well known, is caused by Plasmodium. And that is Plasmodium falciparum. So this is caused by Plasmodium falciparum, which is most common. However, there are other species also, but this is most common. Clear? So this will go with D option. So let us take a look that what are the options that are being matched. So A will go with third option. So this is going to go with this third option over here, which you can see. Then B is going to go with 4 here. Then C is going to go with filariasis is going to go with the first option. C is option is going to go with first option, and D is going to go with the, second option. So here will be the case. All right. So with this, we come to know that option A is the correct answer. This one, entire option. All right. So this is the case. So let's talk about the next question, which is question number 16. And it says that the meiotic division of the secondary oocyte is completed in. All right. So this is a question that is related to the process of oogenesis. All right. And the second meiotic division, if you people remember, then the second meiotic division is actually completed at the time when there is the entry of the sperm inside an ovum. All right. So that is the case. And so if we just look into the options, so it says that it is completed at the time of copulation. So no, this is not an option. After the formation of zygote. No, because this is the fertilization. That means after fertilization products. So this won't be an option. And at the time of fusion of sperm with an ovum. So this is the correct answer. Because if you will look into its explanation, then the second meiotic division is completed at the time of entry of sperm into ova. So you can say as soon as the, or you can say ovum, which is also considered to be as the secondary oocyte. Ovum or secondary oocyte. So as soon as the sperm is going to enter into the secondary oocyte, this will lead into formation of a large ovum along with the tiny polar body, which is the second polar body. So this ultimately results into, formation of a large ovum and second polar body. So two polar bodies are formed in the entire process of oogenesis. First polar body is formed as a result when, if you people just remember it, so the process, how it goes on. So this is the primary oocyte, which is actually present in the, tertiary follicle. All right. So the primary oocyte, which is actually found in the tertiary follicle, this will undergo the process of first meiotic division. So this undergoes first meiotic division, and it ultimately leads into the first polar body and along with the larger secondary oocyte. So this is the first polar body, and this is actually an unequal division. Remember, this is an unequal division, and this is secondary oocyte. And when this secondary oocyte finally undergoes the process of second meiotic division, which is again an unequal division, so this will again lead into formation of, this is first polar body actually, PB, and this will lead into formation of a second polar body and a large ova like this. So this is how the process takes place. All right. So that's why the answer is at the time of fusion of sperm with an ovum. Prior to ovulation, again, this is also not the correct option. So option C is the correct statement. The next question says that by which method was the new breed, that is Hizardale of sheep formed by using Bicaneri eaves and merino rams. So basically Hizardale is a new, a breed of sheep that was actually developed in Punjab. This Hizardale is a new breed of sheep that was actually developed in Punjab. All right. Now, this was actually formed in Punjab or developed in Punjab by crossing the Bicaneri Eve and the Merino rams. Now, in this crossbreeding method, this was actually a process of crossbreeding. So it was developed in Punjab by crossing, you can say, as it has been written over there also, by crossing Eve with merino rams or merino rams, you can say. Clear? So this is an example of crossbreeding. So this is actually a category of crossbreeding where the superior male of one breed is actually mated with the superior female of the another breed. So this is a crossbreeding method. Crossbreeding method in which superior male of one breed all right, are mated with superior female of another breed. Another breed. Clear? This is the case. So with all the options that have been given over here, if we see, so is it mutational breeding? No. Is it a crossbreeding? Yes. So we have seen that yes, it is a crossbreeding method over here. Then interbreeding and outcrossing. Again, these two also are the wrong options. So our correct option comes out to be the crossbreeding method. All right, this is the case. Now, the next question, which is question number 18, it says that the number of substrate level phosphorylations in one turn of citric acid cycles. Now, substrate level phosphorylation basically means that the ATP which is actually being produced from the substrate, that is called substrate level phosphorylation. And if we just look into the citric acid cycle, all right, so the net gain of ATP. So let me tell you about the substrate level phosphorylation first. So substrate level phosphorylation means phosphorylation. Phosphorylation basically means production of ATP. So this substrate level phosphorylation means producing ATP or production of ATP, you can say. Production of ATP from substrate. This is the definition of substrate level phosphorylation. Now in one turn, it is asking about in one turn of the citric acid cycle. So if you look into the citric acid cycle, or if you have seen also, then in one turn of the citric acid cycle, the net gain of ATP from the substrate, or from the substrate level phosphorylation is 1. All right. So if we just write the reaction regarding that, so you can see, and the step which actually converts or produces the substrate level phosphorylation is succinyl CoA in the presence of enzyme succinate thiokinase. This is an enzyme which is going to catalyze the reaction, and this is ultimately going to produce, or you can say convert, GDP, that means guanosine diphosphate, into GTP. All right. So this is the case, and GTP will be further converted into, so this is basically a cycling process, you can say. So either ATP is produced or either GTP is produced. All right, in this process, and only one ATP is being formed over here. Only one, and that too in this reaction only, that is going to convert succinyl CoA into succinate. This is the reaction. So this ultimately results into formation of only one ATP. So how many ATP is being produced through substrate level phosphorylation? The answer will turn out to be 1. All right. So this is the case. Now let's move on to next one, which is 19th question. Now 19th question says you have to choose the correct pair from the following sentences or definitions or functions, you can say. The first statement says that polymerases, it breaks the DNA into fragment. So polymerases doesn't break the DNA into fragment, rather it polymerizes the DNA. All right. So again, this is a wrong statement. So polymerases are involved in polymerization of reactions actually, or DNA polymerization. Polymerases, they are involved in DNA polymerization. So that's why this is an incorrect statement. Clear? Now, option B says nucleases. Now, nucleases are involved in separating the two strands of DNA. No, the two strands of DNA, nucleases are not involved in the separation of the two strands of DNA, because nucleases are actually the kind of restriction enzymes which are going to cut, which are actually going to cut the DNA. So they are actually a category of restriction enzymes that can either be endonucleases or exonucleases. That means making specific cut within the DNA. So this is not going to separate the two strands. The separation is being done by the DNA helicases. So nucleases, the function of nucleases is to act as restriction enzyme. And restriction enzymes are responsible for making cuts in DNA. And cuts at specific site in the DNA. So again, this is also a wrong option. The next question says, statement says that exonucleases. So exonucleases is a category of restriction enzyme. It is one of the category of restriction enzyme that makes cut at specific positions within the DNA. So this is a correct statement. All right, this is going to be the correct statement. Not the correct statement, because this, if you see, then this statement which says that exonucleases may make cuts at specific positions in the DNA. Exonucleases are actually responsible for making cut at the ends of the DNA, not at specific position, but at the ends of DNA. So you can say option C, which is exonucleases. Since this can also be correct, but it has not been mentioned over here. All right. So if you look into the options, so this has been given over here that at specific positions, not within DNA, but at the end of DNA. So that's all. So exonucleases cuts the ends of DNA. Clear? So again, this is also a wrong option. And the D option says ligases. So DNA ligases, they are primarily responsible for joining of the DNA fragments. That means sealing the DNA fragments, you can say. So this is the correct statement. That means it joins DNA fragments. It is responsible for joining the DNA fragments or two DNA molecules, you can say. Clear? So the correct answer turns out to be the option D, which is ligases. This will be the case. All right. Now, let's move on to next question, which is question number 20. So this question says that the infectious stage of plasmodium that enters the human body. So it is talking about, so plasmodium is actually a malarial parasite. All right. This, we know this fact very well. And this is a question from your chapter of human health and disease. So, and the life cycle of the plasmodium has also been, given uh, in detail in that chapter. And so the infectious stage, since it is talking about the infectious stage, so the infectious stage of plasmodium is actually the sporozoites, which enters into the human body in the form of sporozoites through the bite of the infected female Anopheles mosquito. And these sporozoites only ultimately results into chilling effect and high fever, recurring fever, then chilling effect, all those symptoms. So you can say that plasmodium enters the human body so it enters the human body as sporozoites. So this actually enters the human body as sporozoites, which is the infectious state of plasmodium. Through the bite of infected female Anopheles mosquito. So this will be the reason. So if you look into all the options, so sporozoites is the correct option. All right. So this will be the case. Now let's move on to the next question, which is question number 21. Question number 21 says that which of the following is not an attribute of a population? All right. Attribute means character. Now let me tell you one thing, that attribute basically means you have to identify that one of the features, not a feature it is talking about here, not a feature. So attribute basically means character, feature or character. Clear? Now we have also studied this part that population, the one of the characters of population, if you're going to see, the natality is considered to be as one of the characters, because natality is called as your birth rate. So this is a feature of population, natality, which is called as birth rate, is a feature of population. All right. Then again, the second option is mortality. So mortality also is a feature of, you can say, this one, population. So the next is mortality. So mortality is called as death rate. Again, so this is also is an attribute of population. All right. Then the C option says species interaction. Species interaction is not a feature of population because species interaction has not been included in the in the population, because we know that population is a group of organisms of similar species. But in case of species interaction, there will be different species that will be involved. So that's why it doesn't include the it is not included in a feature of population. So this is not a feature of population. You can say this is actually a category of population interaction, but not an attribute. It can be either, intraspecific interaction, or it can be either interspecific interaction, this interaction, species interaction. And the D option says sex ratio. So sex ratio is a feature of population interaction, or is an attribute of population interaction. So an attribute of population. Clear? So out of all the attributes that we have seen, so this species interaction is not an attribute of the population. So the correct answer with respect to this turns out to be option C1. This is the correct answer, not an attribute it is talking about. All right. Now let's move on to next question. Now the next question says that you have to select the correct events that occurs during inspiration. All right. So inspiration, which is also called as inhalation process, that you have to just consider it. So inspiration is actually initiated. Now let me tell you the process, how it goes on. So basically, if we just look into the process of inspiration, which is also considered to be as inhalation process. So inspiration or inhalation. So that is actually initiated, this inspiration or expiration, whether these two processes, all these are actually the combinations, or it is actually based on the principle of the working of the muscles of the rib cage and diaphragm. So this is actually initiated by contraction of diaphragm. So this is going to contract the diaphragm first. The diaphragm muscles, they are going to be contracted, and this will ultimately result into increase in the volume of the thoracic chamber. So this increases the volume of thoracic chamber. And once the volume of thoracic chamber is increased, and that to this volume is increased in the antero-posterior axis, you can say. Antero-posterior axis. All right. So this contraction will ultimately result into this contraction of this, external intercostal muscle is going to increase the volume of the thoracic chamber, also in the dorsal, dorsal ventral axis as well. All right. So if we just look into the statements, the different statements, if you are going to see that, select the correct statements, the correct events that occurs during inspiration. So The first is contraction of diaphragm. So this will be correct. This is going to be the correct option. That means A option will be involved. All right. Then contraction of the external intercostal muscle as well. Again, this is also correct. All right. So apart from the contraction of the diaphragm, there will be the contraction in the external intercostal muscle as well, because this will result into increase in the volume of the thoracic chamber in the dorsoventral axis. So the volume of the thoracic chamber should be increased in the anterior-posterior axis also, and in the dorsoventral axis also. All right. So that's why. So you can just write that while the contraction of external intercostal muscles, which are actually the muscles of your rib cage, costal muscles, increases the volume of the thoracic chamber. So this is going to increase the volume of thoracic chamber, where in the dorsoventral axis. All right. So this will be the case. So how the process of inspiration actually takes place. So this is something like this. If I just draw a sample figure, so you will have an idea. So this gets flattened up like this. And here there will be the muscles that is actually being involved, rib cage muscles over here. So the intercostal muscles, that means the external intercostal muscles, they are also going to get contracted. So these are the rib cage muscles which are going to be up like this. Air will enter from here, all right, and this will go down. That means the contraction of the diaphragm muscles will take. So these are rib cage muscles, and these are the diaphragm muscles. Which are contracted, and this results into increase in volume of thoracic chamber, and so there will be influx of air from outside to inside, because since the volume of the thoracic chamber is increased in the inside part, so this will result into decrease in the pressure in the thoracic chamber, and so the pressure outside in the outside atmosphere will be very high. So this will result into rushing of air, that means oxygen, from outside to inside. So that will be the case. So this is how it goes on. Clear? So pulmonary volume is going to decrease again, no. Then intrapulmonary pressure increases, no. Intrapulmonary pressure is going to decrease actually. In the intrapulmonary pressure means pressure within the lungs. So this is going to decrease, not increase. So the option, if you just look into the options, so A and B are the correct option, and this goes with option D. This is going to go with the option D1, which is A and B. Clear? So this is, so let's move on to the next question. Next question says that according to Robert May, the global species diversity is about. Now, Robert May was the scientist who estimated the global species diversity. So this is a very factual kind of a question that has been asked, you can see, and you just need to remember that fact. So Robert May actually estimated the diversity, the global species diversity, to be about 7 million out of all the options that you can see now. So Robert May estimated global species diversity at about 7 million. So the correct answer is going to be 7 million. However, some extreme estimates, if you're going to look into the extreme estimates, they might range from 20 million to 50 million. But since here it is talking about Robert May, the estimation being given by Robert May, so our correct option turns out to be 7 million, which is option C. All right. Now let's move on to next question, which is question number 24. So next question says that the QRS complex, in a standard ECG represents. So QRS complex basically is a complex which is being observed in a graph that is called as electrocardiogram graph. So electrocardiogram is a graph that represents the movement or the beating of the heartbeat. So if you people just remember it, so this is something, this is going to give an appearance of so the QRS complex, if you are going to see, so the QRS complex comes out to be here. QRS here. This is the QRS complex that comes out to be in the, process of the heartbeat when the graph is being taken out. So this QRS complex indicates the depolarization of the ventricle. So this is going to indicate the depolarization of ventricles. And by counting the number of QRS complex only, the number of heartbeat is being counted. So the number, any deviation from the normal, graph indicates any kind of deformities. So this is related with depolarization of ventricles. And you can say that by counting the number of QRS complex, number of heartbeat is also counted. Heartbeat is also accounted. That means the number of QRX is equivalent to the number of heartbeat. All right. So this is the case. Now, next move on. So the option, out of all the options that you have seen, so the option will be option B. That is the correct answer. All right. Now, the next question is that you have to find out the, incorrect statement. That means the statement which is not correct. So which of the following statement is not correct? So this is a question that has been framed from the insulin that has been, genetically engineered insulin from your biotechnological applications chapter. So basically the first statement says that the proinsulin has an extra peptide which is called as C-peptide. Now you have to identify the not correct statement, which basically means the false statement. All right. So proinsulin has an extra peptide which is called as C-peptide. So this is going to be definitely true. All right. So this statement is true because proinsulin, if you just remember, then proinsulin has an extra stretch, has an extra stretch, and that stretch is of C-peptide only. Of C-peptide apart from A and B. While mature insulin, if you are going to consider the mature insulin, then it has only A and B. A and B peptide, and not the C1. All right, so this is the correct statement, but we have to find out the not correct statement. So the functional insulin has A and B chains that are actually linked together by the, this one, which is actually linked together by the hydrogen bonds. So if you will see the correct statement, the incorrect statement, so this is actually the incorrect statement because it is not linked together by the hydrogen bonds, but the functional insulin A and B, they are actually connected by these two, A and B peptide. They are actually joined or connected by disulfide bridge. So they are connected by disulfide bridge and not by hydrogen bonds. And not by hydrogen bonds. So this is the not correct statement. Genetically engineered insulin is produced in E. coli. True. Again, this is also a correct statement. And in man, insulin is synthesized as a proinsulin. Again, this is also true. So the not correct statement turns out to be this one. That means option B. All right. So this is the correct, not correct statement. Now, the next question is that the transverse section of a plant shows following anatomical features. All right. So transverse section of a plant shows following anatomical features. Anatomical means the internal features, you can say. And these are the features that has been given. Now you have to identify the category of plant and its part. All right. So there are large number of scattered vascular bundles, which is actually surrounded by bundle sheath. The first option. Large conspicuous parenchymatous ground tissue is there. Option B. Option C is vascular bundles conjoined and closed. All right. And then phloem parenchyma is absent. All right. So if you just look into the, different categories, all right, that means the different options that has been given over here. Monocot, dicot, monocot root, dicot stem, dicot root and monocot stem it is talking about. Now the first statement only, the large number of scattered vascular bundle is a characteristic feature of monocot. And apart from that, large number of conspicuous parenchymatous ground tissue is being found. This is also a feature of monocot only. Phloem parenchyma is absent in case of monocot and vascular bundles are conjoined and closed. This is also a feature of monocot. So all these features are actually the features of monocot, you can say. All right. So all features are related to monocot. But now the question arises that whether it belongs to monocotyledon root or monocotyledonous stem. Cotyledonous. Now, all these features are the category or the example of monocot stem. This will be the case. So in case of stem, large number of scattered vascular bundles are found. Then there will be large conspicuous parenchymatous ground tissue is present. Vascular bundles are conjoined and closed, and phloem parenchyma is also absent. So that's why all these features, they belong to monocot stem. So our correct option will be monocotyledonous stem. All right, this is the case. Now, next question. Next question says that you have to select the correct statement with, out of all the statement. That means 3 statements are false and 1 is only the correct statement. So glucagon is associated with hypoglycemia. All right. So glucagon, let me tell you that glucagon is actually a hormone which is released from the pancreas. All right. Islets of Langerhans. Basically, it has alpha cells and beta cells, and these are primarily being produced from the your pancreatic cells. So from the this one alpha cells, and islets of Langerhans from the alpha cells, basically. And this glucagon is actually responsible for increasing the blood glucose level. So that's why glucagon is considered to be as a hyperglycemic hormone. So glucagon increases the blood glucose level. So since this is going to increase the blood glucose level, so known as hyperglycemic hormone. This is called hyperglycemic hormone, not the hypoglycemia. So since this is the hyperglycemic hormone, so here it has been written that it is associated with hypoglycemia again. So then this is a wrong statement. All right. Insulin acts on pancreatic cells and adipocytes. So insulin is basically, insulin, as I already told you, that insulin is being released from the beta cells of the, pancreas. So this is going to act on the pancreatic cells also, and on the, adipocytes are basically the fat containing cells. So this is going to act on the fat containing cells also. Yes. So this is a true statement. Sorry, this is a false statement. Not, it acts on the pancreatic cells, but it doesn't act on the but it doesn't act on the adipocytes. All right. So again, this only acts on the pancreatic cells. So this is the option A. Then option B is insulin only acts on pancreatic cells. Since this is being released from the pancreas only, that's all. Just a second. Yeah. So this is going to act on the adipocytes, and hepatocytes. This will be the case. All right. Now, and this insulin is actually associated with hypoglycemia. So the third statement says that insulin is associated with hyperglycemia. No, insulin is actually responsible for decreasing the blood glucose level. So that's why it is a hypoglycemic hormone. So insulin decreases blood glucose level. So since this decreases blood glucose level, so it is a hypoglycemic hormone. Not the hyperglycemic hormone. So again, this is also a wrong statement. Then the last statement is only left. So it says that glucocorticoids is going to stimulate the gluconeogenesis. So again, this is a correct statement. because these glucocorticoids, since they will promote the process of gluconeogenesis, that means they will increase the blood glucose level or blood sugar level. Gluconeogenesis basically means formation of glucose. So this is a correct statement, that glucocorticoids stimulates gluconeogenesis and so increases the blood glucose level, because they will stimulate the formation of glucose in the blood. That's all. All right. So this is the case. So with this, we have seen that the correct option turns out to be option B. All right. Now let's move on to the next question, which says that Montreal Protocol was signed in 1987 for the control of. Now, Montreal Protocol is one such protocol that was actually signed in 16th September. This is actually, you can say, has been signed for the control of the emission of the ozone depleting substances. However, this protocol was, it came into force, in 1st January 1989. So you can say that Montreal Protocol, this is again a factual kind of a question that you need to know. So this Montreal Protocol which was signed in 16th September 1987. And that's why this is also considered to be as Ozone Day, no? Which is also known as Ozone Day. So for controlling the emission of ODS. ODS means ozone depleting substances. That means this one. All right. This will be the case. So it was actually aimed in, stopping the production also, as well as stopping the import also, the ozone depleting, import of the ozone depleting substances as well. So that's why the correct option is going to be emission of ozone depleting substances. All right. Now, the next question is that you have to match the column with the respective and you have to match the following columns and select the correct option that are found in them. You have to do that part. So let us look into the column first and then column second. So this is basically a question from your animal kingdom Animalia, and the categories of Pisces. It has been given over here, which you can see. So this is the case. Now, if we just look into the questions. So 6, the first option says that 6 to column first. That means this one. Then 6 to 15 pairs of gill slits. All right. So 6 to 15 pairs of gill slits out of all the options that you can see, it matches up with cyclostomes actually. So gill slits are found in the cyclostomes. All right. Cyclostomes because they have an elongated body bearing 6 to 15 pairs of gill slits for the purpose of respiration. So you can just write that, we can just write down here the sentence possible, that cyclostomes have an elongated body with 6 to 15 pairs of gill slits. And these gill slits, they are actually responsible for respiration. All right. So it will go with, the first option will go with this one. A. All right. Now, air bladder. That means if you no, before air bladder, we will see into the heteroserdal, caudal fin if you're going to see. Now, heteroserdal caudal fin is basically present in the members of the class chondrich thighs. Chondrich thighs, this one, that means this one. So this category of fin is actually found in chondrich thighs. So this will go with B option. You can just write that this is one of the characteristic feature actually. So heteroserdal caudal fin. Heteroserdal caudal fin is present in members of class chondrich thighs. All right. So it is going to match with chondrich thighs. Now the next is about the air bladder. So air bladder is actually found in bony fishes. So there are two categories of fishes that you have seen in your course. One is cartilaginous fishes and the other is the bony fishes. So this air bladder is a characteristic feature of ostrich thighs, which are actually the bony fishes, which is this one. All right. So this is going to go with the C option. And the function, let me tell you the function of the air bladder is that air bladder actually helps in regulating the buoyancy, of the fishes, so that they doesn't have to swim constantly, in order to avoid sinking deep inside the, seas or oceans, you can say. So this is relatable to, you can say, air bladder is present in bony fishes. So air bladder is present in bony fishes belonging to class ostrich thighs. That regulates buoyancy. All right, this is the case. The D option is left with only one option, and that is poison sting. Poison sting is found in case of trigon, which is a cartilaginous, which is a category of cartilaginous fish. This is left with D option. Right? So poison sting, is found in trigon. which is a cartilaginous fish. All right, so this is the case. So let us look into the options now. What are the different options that are available? So A is going to match up with 2. A is going to match up with 2. Then here, this one. A is going to match up with 2 over here, this one. Then B is going to match up with 3, this one. C with 4, and D is going to match up with 1 over here. Right. So option 4, that means D option is the correct option. All right. So this is the case. Now let's move on to the next question, which is question number 30. Now question number 30 says that you have to identify the wrong statement with regard to the restriction enzymes. All right. So wrong statement, you have to identify it. So restriction enzymes are basically, let me tell you about restriction enzymes. So restriction enzymes are one such enzymes which cuts the DNA at specific locations. It can be either endonucleases, or it can be either exonucleases. So there are basically 2 categories, all right? So the first statement says that they cut the strand of DNA at palindromic sites. Yes, this is the correct statement, because palindromic sites, if you like remember, palindromic sites are those sites which you will read from, either from the right side or from the left side. It will remain the same, like Malayalam. All right. So yes, this is a correct statement that restriction enzymes, they cut the strand of DNA by seeing the palindromic sites. So let me tell you about palindromic sites. Palindromic sites. Like Malayalam, if you see. So if you will read from here or you will read it from here, both will remain the same. So these are called actually palindromic sites. All right. So this is the correct statement. So they are useful in genetic engineering. Again, this is also a correct statement, because without restriction enzymes, their usefulness, the cutting process, since I told you that restriction enzymes are those enzymes which cuts the DNA at specific sites. Cuts DNA at specific positions. Remember, I'm including the term specific. All right, so that's why. So that's why they're very useful in genetic engineering. True. Sticky ends can be joined by using the DNA ligases. All right. So here the sticky okay. So here it has been written that sticky ends can be joined by using the DNA ligases. So here, if you will see that sticky ends are actually the ends, which actually contains any kind of free or hanging or any unpaired nitrogen bases. So if I have to just write about the sticky ends, then what are actually sticky ends? Let me tell you. So sticky ends are the ends that contains free or hanging, you can say, or any unpaired nitrogenous bases, or unpaired nitrogenous bases, which actually can pair to their complementary bases that is present on the other DNA segment. And here so you can just write that which, yeah, so which can pair to complementary bases. Complementary means in accordance to. That means whatever the bases that will be, you know, in complementation with that of the unpaired nitrogenous bases that will be found. So complementary to the bases present on other DNA segment. All right, that is required to create a kind of a recombinant DNA. So you can say that that is required to create the recombinant DNA. So a ligase is actually required in absence of the sticky ends to join the two DNA segments with one another. All right. So with if there is an absence of sticky ends, then only the ligases can be used. So a ligase is required in absence, remember. In absence of just a second. In absence of sticky ends to join the two segments of DNA. All right. So this is the case. All right. So again, so this is the, actually you can say that this is the wrong state, because these DNA ligases can only function if there is absence of sticky ends. All right. So this is the wrong statement. And the last statement says that each restriction enzyme functions by inspecting the length of a DNA sequence. Again, this is going to be the correct answer. All right, this is also the correct answer. So the wrong statement with regard to the restriction enzyme turns out to be the option C. This one. All right, so this is the case. So let's talk about the next question, which is question number 31. So question number 31 says that you have to match the column with respect to the process of the meiosis. All right. So this question is related to the chapter of your cell cycle and cell division. And, the stages are zygotine, pectine, diplotin, and dikinesis. So all these are the stages, or you can say the phases of the prophase 1 of the meiosis 1. All right. So if we just match out with the respective, options that has been given over here, then this zygotine, the very first important option, just a moment. So zygotine is actually related to the process of synapses. So if we just write the sequence, these are all the processes. So, phases, or you can say stages of prophase 1 of meiosis 1, then the sequence comes out to be as leptotene, then there is zygotine, then pectine, then diplotene and dikinesis. So diplotene, and then comes out to be dikinesis. Kinesis. All right, so this is the case. So this is the sequence. All right, so here basically this zygotine, that means this zygotine is related with the process of synapses or bivalent formation, you can say. So we can correlate the zygotine with synapses, which is usually, synapses is basically a process in which the bivalent chromosomes are formed. This basically results into bivalent formation. So it's bivalent formation or tetrad formation, you can say. All right. So this is related to option A. Clear? Then pectine is related to the process of crossing over. All right. So B is relatable to option 3. So this will go with 3. So B, that means pectine is related to crossover. So crossing over is basically a phenomena in which the exchange of the genetic material takes place between the two homologous chromosomes. All right. And one chromosome comes from the mother and the other comes from the father. So this is the case. So this is the crossing over. Then the third option, which is C, diplotene, is related with chiasma formation. So this one. Chiasma is basically the product of crossing over, which we say. So diplotene is related to chiasma or chiasmata formation. And the last option which is left is dikinesis, which is related with the terminalization of chiasma. So this is D1. All right. So this is dikinesis. This is relatable to terminalization. So terminalization of chiasmata. So this is the case. So if we just correlate with the options, so you can see that A stands with 4. That means this one. All right. Then B is going to stand with, third option. So B is going to stand with third option. That is this one. Then C stands with 2. C stands with 2. Then D stands with 1. That means this one. So our correct option turns out to be option A here. All right. So this is the case. Now, let's move on to the next question, 32. So in question number 32, it is asking about that the cuboidal epithelium with the brush border of microvilli that is found in. All right. So this is one of cuboidal epithelium is one of the categories of the epithelial tissue basically, And this cuboidal epithelium having the presence of microvilli is usually found in, out of all the options that you can see. So this is usually found in the PCT of the proximal convoluted tubule of the nephron. All right. So that will be the answer. So the answer will be that cuboidal epithelium, cuboidal epithelium with brush border of microvilli is found in proximal convoluted tubules, which is shortly termed as PCT of nephron. So nephron is basically the functional unit of kidney, nephron. All right. So this will be the answer. So that means option B is the correct answer. Clear? So this will be the case. Now, let's move on to the next question. So this question says that name the plant growth regulator. You have to name the plant growth regulator, which upon spraying on the sugarcane crop increases the length of stem and thus increasing the yield of the sugarcane crop. All right. So here it is talking about a hormone, basically, that is responsible for increasing the length of the stem. So out of all the hormone, categories, if you will see, so we know that ethylene is a gaseous plant hormone. Abscisic acid while abscisic acid, cytokinin and gibberellin, they have other mode of actions. But here ethylene won't be an option because this is a gaseous plant hormone actually. All right. So ethylene can't be an option because it is a gaseous PGR. So you can just write that ethylene cannot be an option because it is a gaseous PGR. All right. So PGR, plant growth regulator. Abscisic acid is basically, again, abscisic acid also won't be an option. The reason for this is because abscisic acid is it is a plant growth regulator, but it is a negative plant growth regulator. It doesn't promote plant growth, rather it stops the process of the plant growth during extreme condition. All right, so this is actually a plant growth inhibitor. So ABA is one such hormone which is a plant growth inhibitor but not promoter. So this is a category of plant growth inhibitor, and somewhere this ethylene also is included partially in the plant growth inhibitor PGI or plant growth. It has some roles in plant growth promoter and some roles as plant growth inhibitor. So that's why that is also the option that we, that is not the correct option. So the left out options are cytokinin and gibberellin. So out of these two options, cytokinin is basically a cell division promoting, hormone. So again, this is also not the correct option. This is a PGR which actually targets on, promoting the cell division. So this is actually a cell division promoting hormone, you can say. So again, cytokinin is again not an option. So the only option left out is the gibberellin. All right, because gibberellin, spraying of gibberellin, in the sugarcane crop, that ultimately results into increase in the length of the stem, and because of that, around 20 tons of 20 tons per acre of the sugarcane yield is increased because of gibberellin. So this is the, because this gibberellin, it is actually responsible for internodal elongation, and for that reason only this is responsible. So gibberellin basically is the correct answer. So gibberellin, which is also called as GAs, acts in internodal. So its main site of action is in the internodal elongation. So it acts in the internodal elongation and spraying sugarcane crop with GA increases the length of stem. So this actually increases the length of stem, thereby increasing its productivity also, increasing yield by as much as 20 tons per acre, because it actually, the main target site is the internodes only. So for that reason, they are responsible for increasing the yield of the sugarcane crop. So the correct answer is gibberellin. All right. Now, next question. You have to match the column with respect to the correct option. All right. Okay. So basically, the first option that is given is the BT cotton. All right. So BT cotton is relatable to your Bacillus thuringiensis, because BT stands for Bacillus thuringiensis, and this is one such bacterium that has been widely used in the biotechnological experiments for producing disease resistant, disease resistant or pest resistant or insect resistant crops. All right. So you can say that the first option A will be belonging to option 4. So in BT cotton, the gene BT, this gene BT has been isolated from Bacillus thuringiensis only. The gene BT is isolated from Bacillus thuringiensis. It's thuringiensis. All right, so that's why the option turns out to be 4. Now then there is adenosine deaminase deficiency, which is also called as ADA deficiency. ADA deficiency is actually related, if there is a deficiency in this enzyme, so this actually and directly affects the cellular defense in the body. All right, and this is this, regarding with this deficiency, the first clinical gene therapy was performed in 1990 to a 4-year-old girl having the ADA deficiency problem. So ADA deficiency is relatable to option this one, number 2. So this one. So we can just write that the first clinical gene therapy was given in 1990 to a 4-year old girl with ADA deficiency, which is adenosine deaminase deficiency, and this is primarily related with cellular defense. ADA provides cellular defense in the body. All right, so this will be the case. Now, option C says RNAi. RNAi basically stands for RNA interference, you can say. So basically here, here there is a confusion in between the option actually. So we can say, just a moment. So this ADA, actually there is a confusion with this, since this ADA is also responsible for immunity in the body. So there has been confusion with these two options. So actually this RNAi is correlatively, this RNAi over here is correctly related with this one. That means cellular defense. And this is related with gene therapy. All right. So here it has been written gene therapy. I just missed it out. So this one, B is relatable to one option over here. Here, this will be B. All right. And here will be cellular defense. RNAi basically stands for RNA interference, where there is, cleavage or destroying of the viral mRNAs that enters into the body. So this is a method of cellular defense actually. So here will be C option. So you can just write that RNAi which is also called as RNA interference. Clear? Takes place, and this is a method which usually takes place in all the eukaryotic organisms. It doesn't happen in case of prokaryotic organisms as a method of cellular defense. So takes place in all eukaryotic organisms as a method of cellular defense. So this will be the correct answer. And the last one which is left is the PCR, and we all know that PCR, polymerase chain reaction, this is one such reaction which is being used, or you can say it is one such method which has now been used for detecting the HIV infection in the patients. All right, so the option D will be over here. This means PCR is used to detect HIV in suspected AIDS patient. All right, this is the case. Now let's match up all the options, given options. So if we just match up, then A will go with 4. A is going to go with 4, that means this one. Then B is going to go with 1, gene therapy, ADA deficiency. Then RNAi will go with cellular defense, option 2. And D will go with 3 option, which is detection of HIV infection. All right. So out of all the 4 options, D option is the correct option that you can see over here. All right, this is the case. Now, let's move on to next question. Now, the next question says that the whole ovary is half inferior in. So ovary is half inferior basically means that the ovary is in perigynous condition. There are basically three kinds of conditions where the ovary can be found in a flower with respect to the position of the ovary when we determine. It can be hypogynous, epigynous and perigynous. So ovary is half inferior. This primarily means that it is talking about perigynous flower. Perigynous flower. That means where the ovary is in equivalent position with the other whorls of the flower. All are equivalent in position. That means the stamen also, the petal also, the sepal also, and the ovary also. So it is talking about the perigynous flower, and the examples of perigynous flowers includes plum, peach, rose. These are the examples. So it includes, the example includes rose, plum, peach, all these things. So as you can see, out of all the options, the options turn comes out to be as option C, which is plum. All right. So this is the case. Next. Question number 36 says that which one of the following is the most abundant protein in the animals? All right. So most abundant, so this is a question of your biomolecules chapter. So which one of the following is the most abundant protein in animals? So in animals, if you just talk about, so in animals, it is the collagen that has been straightforwardly given in your NCERT also, that it is the collagen which is the most abundant protein in the animals. So collagen is the most abundant protein in animal world. While if you just consider the entire biosphere, so in the entire biosphere, the most abundant protein is considered to be as rubisco. It's rubisco. Just a second. Rubisco, which is ribulose, bisphosphate, carboxylase, and oxygenase. So this is the most abundant protein in the whole biosphere. All right. So with this, the correct option is option A. All right. Now, so let's move on to the next question. So this question says that if the distance between two consecutive base pairs is 0.34 nanometer and the total number of base pairs of a DNA double helix in a typical mammalian cell is 6.6 into 10 to the power 9 base pairs, then the length of the DNA is approximately. So how we are going to calculate it? All right. So length of the DNA it is talking about. So if the distance between two base pairs in the DNA is 0.34 nanometers, so first we are going to convert it into, meters. So let us just solve it. So distance between two base pairs in DNA helix. So this is given as 0.34 nanometer. And if we just convert it into meters, so it will turn out to be as 0.34 into 10 to the power inverse 9 meters. This is in meters. All right. And what about the total number of base pairs it has been given? It has been given over here 6.6 into 10 to the power 9 base pair. So total number of base pairs is 6.6 into 10 to the power 9 base pair that has been given over here. All right. So for calculating the length of the DNA, we need to multiply these two factors. All right. That means the total number of base pairs that are available in the DNA along with the distance between the base pairs in the DNA helix. So if you just multiply the two factors to, this one, two facts, so we will get the entire length of the DNA. So therefore, you can say the length of DNA turns out to be as 0.34 into 10 to the power minus 9, and this is in meters actually. All right, so this is in meters into 6.6 into 10 to the power 9 base pair. And if you just calculate it, if you're going to calculate these two, all right, so that will give you a measurement of around, all right, this with this and this with this one. So this will give you a measurement of around 2.2 meters, like this. All right. So that's why the answer will be option B, which is 2.2 meters over here. Clear? So this is the 37th question. Now, let's move on to next question. 38. So the first phase of translation it is talking about here the first phase of translation. So options given are recognition of DNA molecule, amino acylation of tRNA, recognition of an anticodon, then binding of mRNA to the ribosome. All right. So basically the first phase of tRNA involves not the recognition of the DNA molecule, actually, you can say. Recognition of an anticodon is done later on. This is actually done later on. This is a later phase, late feature, you can say. And binding of mRNA, again, it is also a later stage feature. So it is the very first important phase of translation is to charge the tRNA, that means transfer RNA, with the amino acids, which is called as the amino acylation of tRNA. So you can say the first phase of translation involves activation of amino acid. Activation of amino acid in the presence of the ATP, which is linked to their cognate tRNA. So activation of amino acid in presence of ATP, because this is an energy requiring process, and linked to their cognate tRNA, because they will be having their loops cognate tRNA. All right. So this process, this entire process of activating the amino acid is actually called as charging of tRNA or the amino acylation of tRNA. So this is also called charging or amino acylation of tRNA. All right. So this will be the case. And so the so the correct answer here will be the option number B, which you can see. All right. Now, this won't be an option and this won't again be an option. Now, the next question is that which of the following hormone levels will cause release of ovum, which means ovulation, from the Graafian follicle. All right. So ovulation process is the third phase of the menstrual cycle, and for the release of the ovum from the ovulation, if you people remember, then the options given are high concentration of progesterone, low concentration of LH, low concentration of FSH, and high concentration of estrogen. All right. So this, the correct answer for this, process is basically that ovulation is one such process that which involves the rupturing is the rupturing of the ovary, or rupturing of the ovary, or you can say rupturing of the Graafian follicle that actually releases the ovum. And this, this one, your ovulation is actually induced because of a surge in the LH. That means there is a high concentration of LH. If there will be, but here as the option has been given, there is low concentration of LH, so this won't be an option actually. But when you will see that high level of estrogen will actually this one, that means this option, if high level of estrogen will be there, because before to the process of ovulation, if you people remember, then comes the process of follicular phase. And in case of your follicular phase, there is an advancement in the level, or there is an increase in the level of estrogen and progesterone. Estrogen, LH and FSH are being increased, and along with estrogen is also slightly being increased in amount. All right. So once this estrogen will be high, they will give a positive feedback to the anterior pituitary for the release of LH. And then LH, FSH, and estrogen, when they are peak, they will be responsible for the process of ovulation. So correct answer here will be high concentration of estrogen. And what will be the explanation? So for ovulation, you can say, a high level of estrogen. And regarding this option, first I'll tell you that regarding the options of high concentration of progesterone, this has nothing to do with the ovulation process, because this is a pregnancy hormone. So it has nothing to do with the, your ovulation process. So this won't be an option and this won't be an option. Again, low concentration of LH won't also be there. FSH concentration should actually be high for, for inducing the process of ovulation. All right. So once there is high level in the estrogen, this will send positive feedback to the anterior pituitary. To the anterior pituitary for the release of LH hormone. LH, which is luteinizing hormone. All right. And once the LH hormone is increased during the peak of the cycle, so LH level starts increasing during follicular phase. So during the follicular phase, its level will start increasing and will attain peak. Peak during mid-cycle. Mid of menstrual cycle, you can say. Mid means usually 13th to 14th day. Menstrual cycle leading to LH surge. LH surge. And once the LH surge is being found,
sorry, I don't know that one.
and so once the LH surge is being found, this will ultimately result into the rupturing of the Graafian follicle, releasing ova. So this ultimately leads to ovulation. All right. So the correct answer will be option number D. So this is the explanation that you have seen. All right. Now moving on to next question. Now flippers of penguin and dolphins, they are the example of, if you're going to see. So flippers of penguin and dolphin, they represent an example of, they are actually analogous organs. All right, let me tell you that they are actually the analogous organs, and analogous organs are those organs that are the result of convergent evolution. All right, so that's why here, out of all the options that you can see, industrial melanism won't be an answer, natural selection, adaptive radiation, no. These are because these options, they are extreme in this example. So flippers, you can say that flippers of penguins and dolphins flippers of penguins and dolphins are analogous organs. Analogous organs are basically those organs which are, structurally different but functionally similar. All right. So they might have been, they might be having their, different point of origin. That means they might be structurally different, but their function is similar, which means that flippers in penguins and flippers in dolphins, they both are having the same function, and that too, it helps in swimming, in case of penguins and dolphins. So that is called as analogous organs. Logous organs. So analogous organs always leads to convergent evolution, while homologous organs results into divergent evolution. All right. So analogous organs are the result of convergent evolution. All right. So the correct answer will be option A, which you can see over here. All right. Now moving on to next question, which is question number 41. Now you have to identify the wrong statement with reference to the gene I that controls the ABO blood grouping. All right. So you have to identify the wrong statement. Now, it says, the first statement says that a person will have only two of the three alleles. So remember that you have to identify the wrong statement over here. That means one statement is wrong and the other three are similar. That means correct. So a person will have only two of the three alleles. So this is definitely the correct statement, because out of C, it is usually having gene I has, which controls the ABO blood grouping. It has basically three different alleles, you know, it has three different alleles that controls blood grouping. All right. This is IA, then IB, and I. So this is the case. All right. So again, this is the correct statement. This is actually the correct statement. Now, gene IA and IB are present together. They expresses the same type of sugar. So basically when the gene, or you can rather say when the alleles IA and IB, they produces a slightly different form of sugar, while the allele I does not have any kind of a sugar. This one. These two alleles, they are going to produce sugar. But if there is presence of this one, then this doesn't produces any sugar. Doesn't produces any sugar. All right. So here it says, the second statement says that when IA and IB, they are present together, they are going to express the same type of sugar. See, when both will be present, then, then there will be no expression of any sugar. That means IA and IB, when they are present together. So this is actually the incorrect statement. This is actually the incorrect statement. That means the wrong statement, you can say. All right. Then allele I does not produces any sugar, just as I already told you that it doesn't produces any sugar. So this is also the correct statement. And the gene I has three alleles. Yes, the gene I is having its three different alleles, which is IA, IB, and I. So this is also correct. So the incorrect statement or the wrong statement turns out to be option number B over here. That is the case. All right. So when IB, IA and IB, they are present together, they express the same type of sugar? No. When they are present together, so they actually produces a slightly different form of sugar from one another. All right. So you can say that the alleles IA and IB produces a slightly different form of sugar. Not exactly same form of sugar. Clear? This will be the case. Next question. Now you have to select the correct option, including all the sexually transmitted diseases. So options regarding all the sexually transmitted diseases has to be included over here if you're going to see. So in the very first important option, as you can see, that gonorrhea, malaria, genital herpes, gonorrhea, malaria, and genital herpes. So out of all these three, option A, if you're going to see, then malaria is actually caused by plasmodium. This is a protozoan disease, so that is not included in case of your genital herpes. So if you just, it is gonorrhea, then malaria, then genital herpes. All right. So this is an STD, sexually transmitted disease. This is an STD, but this is a protozoan disease actually, which is caused by Plasmodium. All right. So this won't be an option over here. All right. Now let us take a look into the second option, which is AIDS, malaria, and filaria. So AIDS, malaria, as I already told you, is a protozoan disease, and filaria. So filaria is basically a helminthic disease, filariasis, which I tell you. So this is a helminthic disease, which is actually caused by worms. This is a helminthic disease, which is not a sexually transmitted disease. So this is an STD. However, these two are not. All right. So again, this won't be an option over here. Then the third next option, that means C option, tells you cancer, AIDS, and syphilis. So cancer, AIDS, and syphilis. Syphilis. So here, let me tell you that cancer is not a sexually transmitted disease. So this is not an STD actually. All right. AIDS is a STD, category of STD, and this is also STD. So again, this won't be the correct option. All right. And option D, which is gonorrhea, syphilis, and genital herpes. So gonorrhea, just a moment. It's gonorrhea, syphilis, and herpes. Genital herpes, basically. Herpes. So this is the correct combination of the sexually transmitted disease, because this gonorrhea is actually caused by, this is actually caused by a bacterium which is called as Neisseria gonorrhea. So this is caused by Neisseria gonorrhea. Then this syphilis is caused by Treponema pallidum. So this is caused by Treponema pallidum. This is again a bacterium. Just a second. It's pallidum. And herpes, genital herpes, is basically a category of STD which is caused by type 2 herpes simplex virus. So this is caused by HSV, which is also known as herpes simplex virus over here. So out of all the combinations that we have seen, so the correct is option number D over here. All right. Now, moving on to next question, which is question number 43. So question number 43 says that you have to match the column and select the correct option. Again, that's the match the column that has been given over here. So there is on the column first, there are various categories of WBCs has been given, and regarding to that, in column 2, their responses are being given. All right. So the first is basically your this one, that is going to be the eosinophils, option A, which is your eosinophils it's talking about. So eosinophils, they are actually associated with allergic reactions. Eosinophils are related with allergic reactions, And the enzymes which are being released during the allergic reactions are usually the histamine and all those things, histamines and other destructive enzymes. So this is going to match up with this one. All right. So eosinophils are associated with allergic reactions. All right. So eosinophils are actually associated with allergic reactions, and so they releases histaminase and destructive enzymes, and releases histaminase and destructive enzymes. Clear? Now, the option B is basophil. So basophil is basically related with the release of the granules that that regulates the granules that actually contains histamine, because basophil secretes those histamine, serotonin, heparin, etc. that is involved in case of all the inflammatory reactions. All right. So B is going to match up with this one. This one. So you can say that basophils secretes histamine, then serotonin, heparin, etc., and are involved in inflammatory reactions. All right. So this is going to match up with option B. Now, next option is neutrophils. Next is neutrophils. Now, neutrophils, they are actually, involved in case of the process called as, you can say, phagocytosis. Basically, these are phagocytic cells because they produces those B lymphocytes and T lymphocytes that are, that is related with lymphocytes I'm talking about. So these neutrophils, they are actually related to the, to the process of the this one, which is considered to be as the phagocytosis process. All right. So we can say that these are phagocytic cells. So neutrophil is going to match up with the phagocytosis. That means C over here. So neutrophils are phagocytic in nature. Phagocytosis basically is a process in which there is engulfing of the foreign particle or any of the, material that enters inside the cell unknowingly, or, by one or the other way. So that is basically these neutrophils. What they do is that they directly engulf all those foreign particles, or, or those, damage secreting cells or something. So they are going to engulf that. Phagocytic cells. All right. Now the last is left with lymphocytes. So lymphocytes are related with the immune responses. So lymphocytes are basically two categories. It can be either B lymphocyte or it can be either T lymphocytes. So lymphocytes, that can be either B and T. They are responsible for immune responses of the body. Immune responses of the body. All right. So if you just look into the options, so let us take a look where does it matches. So A is going to go with option 3. So A is it with 3. Then B will go with 4. C will go with second. And D will go with first option. So our correct option turns out to be option D over here. All right, this is the case. Now, the next question is, again, you have to match the trophic levels regarding to the different species that are actually found in case of the grassland ecosystem, if you tell me. So again, if we consider a grassland ecosystem, so grassland ecosystem is actually a terrestrial ecosystem and it includes various kinds of trophic levels. So you have to match up with the trophic level actually. So we can say that grassland ecosystem is a terrestrial ecosystem. All right. And if we just look into the various kinds of trophic levels which are found in them. So the first trophic level, if we consider first trophic level, so we know that the first trophic levels are occupied by grasses only. So this will be occupied by grass. So first trophic level, that means first trophic level, option C will go with grass. So this is with option C. All right. Now, if you look with the second trophic level, so grass will be eaten by herbivores. All right. And regarding all the options that you can see, there you can see only one option to act as a herbivore, and that is rabbit. So this will go with the second trophic level. So this means B over here. Clear? So this will be rabbit. Then again, the third trophic level. The third trophic level will be represented by someone who is a carnivore. All right. Yeah. So it comes out to be as crow. Third trophic level will be represented by crow. So this will go with D option actually. All right. So third trophic level will be crow. And the fourth trophic level, which is the last trophic level of the food chain, will be represented by vulture. So usually vulture, eagle, all these species, they occupy the, last section of the trophic level only. So this is the case. So if you're going to match up with the column, so A will go with, this will go with A option actually. So A option will go with 2. A will be with 2. Then B with 3. B will go with 3 option. Then C with 4. And the D with 1 option. So the correct answer will be option D over here. Here will be the correct answer. All right. So this is how you have to do it. Now, the next question is that in the light reaction, if we have to consider, then plastoquinone facilitates the transfer of electrons from, all right. So it is talking about the light reaction, and light reaction, this is talking about plastoquinone. So this is basically, an electron carrier which is involved in the non-cyclic electron transport process. All right. So you can say we can first have a look into the explanation first, and then we will find out that where it is going to transfer it. All right. So you might have heard that once the electron gets excited from the PS2. So how the process goes on, if I just tell you, So this is something that it is a kind of a Z scheme process that goes on. So here is the PS2, which is going to get excited over here due to light. And this PS2 is also associated with splitting of water that is going to release electron also. All right, so this excitation of electron will result into the excitation of the photosystem also over here. And again, it is going to pass on the electrons through various carriers. And then in the center, there is a cytochrome B6F complex. So B6F complex is there, which acts as the communicator, you can say. That means the linking complex between PS2 and PS1. Then again, this will go like this, and it will pass on the electron to PS2. Sorry, PS1 over here. Then again, this will be excited by light over here. And here will be PS1. So this is a kind of a Z scheme flow, which you can see. This is actually called as Z scheme, which is the non-cyclic flow of electron in the light reaction. All right. Now here the electron carriers are actually involved. So you have to see what is the role of the plastoquinone over here. Plastoquinone is actually going to facilitate the transfer of electron options. Various options have been given over here, which you can see where it is going to transfer. So plastoquinone somewhere lies over here. All right, this is one such electron carrier which is going to carry the electron over here. All right, so if you just look into the figure, so you will have an idea that plastoquinone is actually carrying the electron once the excitation takes place. Here basically the red light falls upon. So here will be the red light that will be striking upon. Red light. And here will be far red light that will be striking upon. So here will be far red light, which is usually having a wavelength of beyond 680 nanometer. That means 700 nanometer. And this is of this PS2 red light is having a nanometer of 680. So this is far red light. All right. So if you can just look into the figure, you will have a clear-cut idea that plastoquinone is somewhere lie over here. All right, this here at this point. So this is going to transfer the electron from PS2 to the cytochrome B6F complex, which is a linking agent between PS2 and PS1. All right. So is it cytochrome B6F complex to PS1? No. Is it PS1 to NADP? No, because NADP somewhere lies over here if you're going to see. PS1 to ATP synthase? No. Then PS2 to cytochrome B6F complex? Yes. This is the case, which you can see over here. All right. So our correct answer will be PS2. It is going to transfer the electrons from PS2 to cytochrome B6F complex like this. All right. So this is the case.